Okay, folks, let's get started. T-1 and counting, right? Or 2, I guess, depending on how you count things. If you count class days or actual days. So, examine here on Friday at noon. And we'll do like we did before. Please spread yourselves out. When you run out of seats, just set every other one, then sit up front. And we will deal with that. I have scheduled a review session. It will be on tomorrow evening at 5:15 in ALS 4001. And I will videotape it and obviously get the videos posted as quickly as I can. We're going to have a decent amount of time today to talk about questions that you have and so forth. So today will serve as sort of a mini review session as well. There's not a lot of things I have to say today. So that's what's on tap here. Questions about that? Okay. So last time I spent some time talking about a couple of repair mechanisms. And I just want to briefly remind you about those. The first of which was mismatch repair. And mismatch repair is a repair system that is useful for fixing errors in replication that don't otherwise get fixed. So if something escapes the proofreading mechanism, this system here is good at coming along and fixing those. This system of the 3 that I talk about is the only one where the cell has to make a decision about which strand is the one to repair. And so as I mentioned last time, the cell uses, and again, these are all in E. coli, although we have very similar systems in our cells. In E. coli, they use a methylation signal, and that methylation occurs after all the replication is complete and has been done for a while. So when that happens, the DNA that's been made gets completely replicated. So when we start the replication process, we have the parental strand is completely methylated because it's already been through the methylation process before. The newly synthesized strand, of course, has not been methylated. And as a consequence, the cell the cell can tell which strand is the strand that it's just made. Therefore, the mut-S protein comes along and binds, and this system recognizes which strand to excise, and it makes a cut, excises that, and then fills it back in to make a completely intact strand. At this point, the DNA would be ready to go, and the cell would then methylate this other strand now so that we would have completely methylated DNA, and it would be ready for the next round of replication when that would occur. Okay? So pretty straightforward system. Kind of a neat trick. In humans, we don't methylate. I should point out, we don't methylate the GATC sequence. We actually methylate CG sequences. And it's not completely understood in humans how the CG system works, at least certainly not nearly as clearly as we understand how it works in E. coli. All right, the other system I talked about last time is called nucleotide excision repair. And nucleotide excision repair is a mechanism that's very useful for removing DNA adducts and for thymine dimers. So these are modifications to the DNA that will cause a bulge, a different kind of a bulge than we see with a mismatch. And as a consequence of that, this bulge is recognized. It's quite clear which strand has the problem. This UVR-ABC, which is what we'll just call it UVR-ABC, UVR-ABC system comes along and basically does what the last system did as well, makes a cut in the appropriate strand, excises back, and then DNA polymerase 1 comes along, fills in. And every time when we fill in a gap like that, we not only have to have a DNA polymerase, we also have to have DNA ligase because that last phosphodiester bond is not made by the polymerase. So we had to have a DNA ligase. The previous slide didn't show DNA ligase, but in fact it is also needed for that completing of the process. At this point, the DNA is good to go. Now you might look at this and say, well, Kevin, if I've got this repair system in me, and yes, you have this repair system in you, why should I not go to tanning booths all the time And the answer is, I don't think you want to take a chance on overloading the system that you've got. So there's enough mutation going on in other areas that are problematic that you don't want to increase your chances of that. And anything that escapes this system or overloads this system is going to strongly favor mutation. More mutation, of course, favors cancer. Okay, so those are the two systems I talked about last time. What I want to say a word about now is another system, and on here it's labeled as uracil repair, but it's actually called base excision repair. Okay? So base excision repair is most commonly used to remove uracils from a DNA duplex. Well, how do uracils get there? Remember that in the synthesis of thymidine nucleotides, there was some DUTP made. And though there is the enzyme DUTPase, which is designed to remove the DUTP, some of it does make it into DNA. Well, having U in DNA is not a desirable thing. And as a consequence, a cell has an excision mechanism that allows it to specifically remove U from DNA. Okay? And it does it by using this enzyme called uracil DNA glycosidase. Uracil DNA glycosidase is working in a different way than the other systems that we have seen. So uracil DNA glycosidase is clipping the base out of the duplex. Notice that the phosphodiester bond remains intact, at least for the first step. We've clipped the base off, but the phosphodiester backbone is still intact. After the base has been clipped, then a break in the phosphodiester bond is made, and excision happens as before, and replication to repair it. So base excision repair is specific for bases and allows the cell, most commonly, other things that can be done, but most commonly allows U's to be removed from DNA, as you see here. Okay, so those, and as you could probably guess, when you see a U in DNA, the cell has no difficulty deciding which strand to fix, because obviously the U is already in that strand. So it takes care of that process. Okay. Now, the relevance of DNA repair to human health is not trivial. There are many places and increasing bodies of knowledge that we have about the effects of loss of DNA repair function and its impact on human health. Okay? There are a couple of examples that I will cite. The first of these is HNPCC. HNPCC stands for hereditary non-polyposis colon cancer. I think that's right. HNPCC. And this is the most common type of inherited colon cancer that is found. And it's inherited, meaning that there's a defective gene that is passed from generation to generation. And the defective gene is known. It's a gene that's involved in DNA repair. So not surprisingly, if your DNA repair system is not functioning up to snuff, then you may well be much more prone to mutation. And of course, more mutation translates to more cancer. So HNPCC is very actively studied. There's a professor on campus, Dr. Andrew Burmeyer, who this is actually his main area of research. Shannon? Why is it in colon? I knew you were going to ask that. It's the most common question that we get. I don't have an answer for that. When we think about a repair system that's necessary, it could be that you have higher incidences of other cancers in the body as well, more commonly manifested in the colon. The colon is a place where a lot of things are just shunted out. And as a consequence, that could be that there's more exposure to other mutagens that are there that need repair, and other factors as well. Yes, ma'am? Is there some sort of gene therapy for it? Is there a gene therapy for it? To my knowledge, there's not. The system itself isn't completely understood. And so what Burmeyer's lab does, in fact, is isolate DNA repair enzymes or defective DNA repair enzymes from isolated tumors so they can better understand the role of that in that process. After those are better understood, perhaps something would happen with that. But gene therapy is probably not a direction that will happen, at least in the near future. Yes, sir?
Do they have a DNA test for it, like rapid testing?
That's a very good question. Do they have a DNA test for it? I believe there is a DNA test for some components of that, but I don't know the parameters on it. There are different repair systems that can have problems in causing HNPCC, so whether one test system would work, I don't know that. But I believe there are some, yes. The gene, the most commonly mutated gene, is actually known, so that would make you think there probably is a system. Okay. Another place where DNA repair is implicated is in breast cancer. There's a gene called BRC-A1, and BRC-A1 turns out to have functions in the repair of DNA. Again, I don't have an answer for why it's primarily breast cancer, but again, that is most commonly associated with breast cancer. I know in the case of the BRCA1 that gene is frequently examined in people who have developed breast cancer and also in their siblings and progeny to better understand the likelihood of having breast cancer. There are, I believe, 3 BRCA genes that are now known, and the likelihood of developing breast cancer with mutations in those is fairly high. So that's a concern, obviously. Okay, so DNA repair, again, not surprisingly, is important. If we don't fix those adducts or we don't fix those problems that are in the DNA polymerase will get to those, and then there's real problems. Now, cells have a seemingly, I won't say infinite, but a seemingly very large number of check systems to ensure that they don't have any more problems than necessary. And one of these is the last thing I'll talk about with respect to DNA repair, and this is the protein known as P53. P53 is a protein that has been described as the guardian of the cell. And I hope after I describe it to you that you'll have a better understanding about why we describe it in such a fashion. P53 is a protein, and it's a protein that basically assures quality control of DNA replication. So you saw, for example, in eukaryotic cells that, and by the way, P53 is a eukaryotic protein. It's a protein that's present in every one of our cells of our body. You saw that during the lifetime of a cell, there's a cycle through which cells synthesize DNA and through which they divide, et cetera, et cetera. And that cycle is reproducible. It's like a clock. It goes on and on and on and on. During the synthesis phase of the DNA replication, problems may arise with replication of DNA. Let's say that we have an adduct that can't be removed. Or let's say that we've had damage to a piece of DNA or loss of a piece of DNA. There's some problem that's happened that can't be fixed by the repair system. Should that occur, the replication of that DNA will not complete. And so what P53 does is it has a sensing ability to basically determine if replication has proceeded properly or not. And that's that sensing ability is way beyond what we will talk about here. It's not like a nerve sensing, so don't get that idea. Anyway, if P53 gets the signal that replication has not completed properly, P53 will immediately stop the cell cycle. It will stop the cell cycle, and P53 is a transcription factor, meaning it's a protein that can bind to DNA, and what it will do at that point is it will bind to specific promoters in the genome, and those promoters are for genes to repair DNA. So when the replication does not complete properly, P53 stops the cell cycle. It induces the production of repair proteins, and those repair proteins will then go and attempt to fix the damage with the DNA. Now, it's important to stop the cell cycle because you don't want to be doing all that repair while division and so forth is going on. You can imagine chromosome breakage and some other problems, et cetera, et cetera. Now, the repair proteins go. They didn't get it the first time around. Will they get it the second time around? Maybe they will, maybe they won't. All right? If they get it the second time around, P53 says OK and restarts the cell cycle. If they do not get it the second time around, P53 will induce the cell to commit suicide. Okay? Cellular suicide has a name. It's called apoptosis. It's the most commonly mispronounced name in all of biology. Most people say apoptosis. Okay? And if you want to be a complete idiot, say apoptosis. Okay? There's a we could describe, for example, pneumonia, and if we had something that was related to pneumonia called apopneumonia, we wouldn't say apopneumonia. So for the same reason, the P is silent. As it's silent in pneumonia, the second P is silent. So apoptosis is a cellular suicide. That cellular suicide is induced by p53. Now, cellular suicide is very, very important because if the cell does not commit suicide, then it has a mutation that hasn't been dealt with, and that mutation that hasn't been dealt with now can lead to uncontrollable growth. Let's say we screw up a signaling system because we mutate a protein that's essential for the signaling system. Multicellular organisms have to have this type of built-in control. If we don't have this type of built-in control, then we are much, much more likely to develop a cancer. Okay? So having a cell commit suicide is a very, very important thing. It comes up in a couple of ways with respect to cells. And it is, as I said, an essential function for a multicellular organism. Now, the next question you probably have is, well, what if I mutate p53? What happens? Okay? Well, it turns out that mutation of p53 is strongly correlated with cancer. There's something like 90% of certain types of cancer, including, I believe, colon cancer, that have a mutated p53. Once you mutate p53, now you've taken away the quality control. You're much more likely to have a giant increase in numbers of mutations within that cell, and some of those may lead, again, to uncontrollable growth. So p53 is a very, very critical protein in this process in keeping cells from having excess mutations and getting out of control. Questions on that? Yes? You can't imagine all the what mutating it?
Well, does every cell only have one p53 controlling it?
It has, okay, so there's one p53 gene, yes. Are there multiple proteins made from that gene? There's a few, not a large number. Okay? So, but if you mutate the gene for that p53, then every protein that's made from it is going to be a mutant form, and it's not going to it may not function properly. Okay? Okay. So, it's important to protect our genome. All right. The last thing I want to say about DNA repair is only peripherally related to it, and it is something called the Ames test. Now, the Ames test is a very simple test that was designed to tell us the likelihood with which a compound will cause or favor a mutation. Okay? So with the Ames test, what we're interested in doing is understanding the likelihood a compound will make a mutation. How do I measure that? Well, it turns out some people have come up with some very clever ideas. Bruce Ames was the person who came up with this scheme, and it's a very simple one. Simplicity is good when we're trying to measure something that's fairly complicated. Okay? Well, I need to describe to you how it works, and then I'm going to show you and describe some results for you. All right. When we're looking at mutation, when we're trying to understand mutation, we're trying to see how frequently it happens, right? Well, I've already told you that mutation, even in things that are very error-prone, doesn't happen very often, right? Okay? So if something happens at 1 in 10,000, that's pretty high. Okay? More commonly, we're seeing mutations of 1 in a million, 1 in 100,000, maybe 1 in 10 million. Okay? We have to have a system that's sensitive enough that will allow us to see that one mutation, okay, and not see all the other things that aren't mutated. That's what our aim is. We want to be able to see that one mutation, and we don't want to have to see all the other noise associated with that. That make sense? Okay, so the system I'm going to describe to you then has a way for us to specifically see that rate with which that mutation occurs without all the other noise. All right, so how does it work? Well, there are several ways of doing it. I'm going to describe for you what I hope is a reasonably simple way. Let's imagine I have an E. coli bacterium. And that E. coli bacterium contains within it a plasmid, it's a circular DNA, that carries a gene that gives that E. coli resistance to tetracycline. Tetracycline is an antibiotic. Okay? So if I take that E. coli bacterium and I grow a lot of them up and I plate it on a petri dish with agar, and the agar contains tetracycline, what am I going to see? I'm going to see growth like crazy, right? Okay, so that's not the system I use. I modify that system to make it work the way that I want. All right? Let's say I take that same E. coli bacterium and I make a single mutation in one critical base in that gene that gives tetracycline resistance. Okay? If I make that mutation, that one base, and I studied this and I determined that that mutation causes the cell to not be resistant to tetracycline. Okay? Then if I take that and I plate it on an agar dish with tetracycline, well, first of all, I plate it with no tetracycline, what am I going to see? Complete growth, right? If I put tetracycline on there, what am I going to see? No growth? Well, maybe and maybe not. Okay? What do you mean maybe not? You just told me you mutated the gene. The gene is necessary for tetracycline resistance. How would I get growth? How would I get growth if that happened? What would have to happen in order for me to get growth? What if I mutated the mutant back to what it was originally? Let's say I mutated the G to a T, and so I've got these bacteria that are sitting here, and they've got G mutated to T. As long as G is mutated to T, they're not going to grow on tetracycline. However, if their T mutates back to a G, they're back to the original, bang, they can grow like crazy. Right? Well, the beauty of the system that I've just described to you is that mutation doesn't happen very often. It doesn't happen very often. So if I just took these cells and I plated them out on a plate, and I started out with millions of cells, or maybe billions of cells, if I had a rate of mutation of, let's say, 1 in 100, okay, I'm sorry, a rate of mutation of 1 in a million, I might see a few hundred colonies on that plate. Not a billion, but the ones that I see, I would know arose from a mutational event. Does that make sense? Okay. Questions on that before I go forward? Because I've got to describe the experiment to you in a second. You understand what I've done? Yes?
Would it be more likely that the reverse mutation would happen because that would fit better into the DNA, or like the repair systems?
Okay, would the reverse mutation be more likely because it would fit better in the DNA? Remember that I've already made the mutation and the DNA is perfectly functional. That is, it doesn't make a functional gene, but there's no mismatch as such. We've already, that's already been taken care of. So I've grown these guys up independent of antibiotic. They've already repaired that mutation in their DNA. So they've got a T where they really should have a G. There's no bulge or anything like that. Okay? So it's not more likely to go back. And we don't want it to be more likely to go back. We want to measure the random rate with which it goes back. So that's an important consideration. It's a good question. All right. So I've got this billion cells of E. coli sitting over here that I grew up in the absence of antibiotic, obviously, because if I tried to grow them in tetracycline, I couldn't grow them up in the first place, right? I grow them up in the absence of antibiotic. I've got a billion cells over here, and I've got a billion cells over here. Why do I have two? Well, I'm getting ready to test to see the likelihood, okay, that a mutation is going to happen. One group I don't treat. The other group, I have a theory that the French fries at McDonald's have a substance in them that favors mutation. Not an unreasonable thing. Hello, McDonald's. Not an unreasonable thing. We'd say, okay, so what's the likelihood? I've got this compound isolated. So I take one group of cells, I don't do anything, and the other group of cells, and I mix it with this compound. I take this mixture now, and I put it on plates. Okay? Well, in order to make a colony, cells have to replicate. They have to replicate their DNA. They have to divide. They have to replicate their DNA. And I ask the question, which plate has more cells? If I see roughly equal numbers of cells on the two plates, what would I conclude? the compound had no effect because it didn't affect the base level of mutation that I would normally see just from regular mutation in the cell. On the other hand, if I see a significantly higher number of colonies on the treatment plate, I would say I should stop eating McDonald's French fries, which is probably a good idea in the first place. Okay? Makes sense? All right. So the Ames test relies on simple mutations like that. And there's other ways of doing the same thing, but relies on simple mutations like that where I can easily compare how many here versus how many there. If I see a significant increase, I have to start worrying about that compound. Yes, sir?
It seems like to do that, you're changing the fidelity of the DNA polymerase, but most of the mutagens we're talking about things like thymine adducts or thymine cross-linking adducts, those just stop DNA polymerase and just confuse it rather than changing its fidelity.
Okay, so his question is, it seems like I'm changing the fidelity of DNA replication as opposed to, say, putting adducts in there. Is that what you're saying? All right, but the point is that either way, I will have more mutation. If I put more adducts in there, I've got to rely on every single one of them to get removed properly. And just like I don't want to overload my thymine dimers, so too will I have difficulty with adducts. If my DNA polymerase itself is being attacked by this compound and its fidelity changes so that it doesn't put in G next to C as efficiently as it should, for example, I'll see the same thing. So all this test tells me, it's a very simple test. It doesn't tell me what the mechanism is of the mutation. It simply tells me that this compound over here is favoring mutation compared to no compound at all. Does that make sense? Shannon?
Doesn't the Ames test involve liver enzymes?
Ames test can be done in several ways, actually.
One type of test?
But I don't know what you mean by liver enzymes. The Ames test itself is actually a cellular assay like what I'm describing here. So, okay. Now the figure on the here is not the best one, unfortunately, but it's basically showing you a control plate and another plate where there is a higher growth, basically. Okay, they're actually doing something a little bit different in this experiment than what I've described to you. But suffice it to say, the Ames test works by counting colonies as to how many colonies become resistant. Okay? Okay. So that's the Ames test. Ames test is simple. Ames test is fairly quick. You can do this overnight, for example, and have a very good understanding about the mutation rate of cells. All right, the last thing I want to talk about, and then I will turn this over to some questions, is recombination. Recombination is a phenomenon. It's a natural phenomenon out in nature. It's one of the reasons that we are what we are. Recombination happens when different DNAs swap strands, basically. When DNAs swap strands. Now, I said different, but actually the most common things that are swapping strands are actually related DNAs because that helps the base pairing process, et cetera, et cetera. We can imagine that if I have, let's say, two chromosomes in me, one chromosome number 1 from my mom and one chromosome number 1 from my dad, that they would be very similar in base pairs, but they might not be identical. So if I swap strands, what am I going to see if I swap strands? I'm going to see some bulges, right? What's going to happen when I have those bulges? Well, hopefully I'm going to induce some repair, and that repair is actually going to change the sequence of the resulting thing. Recombination, therefore, gives rise to new DNA sequences that didn't exist before the recombination. I'm going to show you the mechanism of that. It's going to make it very simple, so don't worry too much about that. But recombination gives rise to new sequences that didn't exist before. Now, I raise that issue because it's a very, very important thing for us to consider. We think about, well, I've got one set of chromosomes from mom and one set of chromosomes from dad. So therefore, I am a perfect blend of the two of my parents. And in reality, I'm not. And you're not, and nobody is. Okay? Because recombination happens during the development process. The recombination happened when you were a fertilized egg. It happens in every cell. And as a consequence, you have DNAs that neither of your parents have. A lot? No. A few? Yes. Okay? So recombination gives rise to new DNAs that neither parent originally had. That's important to keep in mind. Okay? Evolutionarily, that process has very high significance because that is one way in which we rapidly diversify genes going from generation to generation. Okay. Well, let's just take a quick look to see what happens in this recombination process. Okay. This shows something of a mechanism in which we have two DNA, this might be two chromosomes that are lined up with each other, and they're very, very similar in sequence, and one strand is actually able to invade the other one. So we see this strand here invading because we've got an excess end hanging off, and no, we won't talk about how we get that end at the moment, but we get invasion. That invasion is not unreasonable because, again, this thing is base paired, and once it starts base pairing, this thing can start zippering its way up because those base pairs fit very nicely. They don't have to be perfect. They can be pretty good. Okay? Well, this goes for a point, and as it zippers up the thing, ultimately it will create a structure called a holiday junction, most commonly. Holiday junctions arise, and this is simply not showing that zippering process. Okay? Holiday junctions arise during recombination, and they basically form a cross-like structure. Holiday was named for the person, holiday structure named for the person who discovered them. Notice his name has two L's in it, not one, like Christmas. Okay? All right. They form, ultimately, a cross-like structure. So you can actually see that cross-like structure here. And depending upon how this cross-like structure resolves itself, what does that mean? Well, I can cut this bond a couple of ways. I can cut it this way. I can cut it this way and remake the duplex. So there's a couple ways I can make that. Depending upon how this guy resolves itself determines what I ultimately end up with in my recombination. Okay? Holiday structures are real things. We can isolate inside of cells. We can actually isolate molecules in the middle of recombination. And when we look at these things, they have a very nice cross-like structure. Okay? Well, obviously the structure has to be resolved. There are some enzymes that are necessary both for the formation of the holiday structure, which we won't go into, and also in the resolution. But as a result of this, we can have some significant swapping of DNAs from one chromosome or one DNA sequence to another. This, I point out, is not totally unlike what we do in the laboratory where we're making recombinant DNAs. In making recombinant DNAs, what we are doing is we're putting together sequences that weren't together originally. And though we're using restriction enzymes and DNA ligases to do that, all right, the point is that we're simply doing a recombination that's happening. Okay? So when you hear the word recombinant DNA, it doesn't have to mean bioterrorism or something like that. It can mean something very innocuous. And certainly in our cells, it means something that's very innocuous. Okay. Nah, we don't talk about that. Okay. So that is the last of what I have to say for the exam. And at this point, we've got about 15 minutes left. So if you would like to, I'd be happy to try to answer any questions you might have. Yes, sir.
Right, so you're talking about the starting point of synthesis of fatty acids. The CoA gets replaced by ACP. When and how does that happen, and how does it go back?
Okay. So during fatty acid synthesis, CoA gets replaced by ACP. How does that happen? Is that your question? And
how and when?
How and when? Okay. So go back to fatty acid synthesis. Basically, all I can say in terms of when is it happens before the synthesis process starts. So once you get out into the cytoplasm, acetyl-CoA and malonyl-CoA, those CoAs will rapidly be replaced by ACPs and converted into the synthesis process. But to go back to that outline, okay? So, fatty acid oxidation, fatty acid synthesis, down here, okay? So, there, we remember first of all that when we have acetyl-CoA, it's from acetyl-CoA that we make malonyl-CoA. that malonyl-CoA is an essential step in that process because that's the 3-carbon intermediate that's necessary. But this conversion occurs on CoA. That's why we call the enzyme name that does this acetyl-CoA carboxylase. It puts an extra carbon onto an acetyl-CoA. After we have this, okay, we will then take and put an ACP on it. We will also put an ACP onto some of the acetyl-CoAs. This is not something that goes to completion, obviously. And as a result, we will have acetyl ACP and malonyl ACP. And that is what is depicted here. Okay? So our starting materials for the overall process are acetyl ACP. We've replaced that CoA with an ACP. Malonyl ACP, again, we've replaced the CoA with an ACP. And we're able to start that process. At this point, we're in that fatty acid synthase complex. That's that wheel that's turning around. Does that answer your question?
Yeah.
I've had several people asking about that ACP, so I know there's a little confusion on that. But that's basically what's up with that. Other questions? So I guess I don't need to do a review session then, right? Everybody's got all their questions answered? Yes, ma'am. Here? Nope. 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 I want you to understand the overall process. So we're adding 2 carbons each time by starting with a 3-carbon compound. We're adding those 2 carbons, and when we add the 3-carbon compound, one of the carbons pops off, so only 2 go in. And then you should know, obviously, the chemical nature of it. That is, we're going backwards the process that we did of fatty acid oxidation. We reduce the ketone to a hydroxyl. We pull water out to make a double bond. We add hydrogens across that double bond, and then we go through another round. Make sense? Yes, Jen?
Can you do a general overview of the fatty acid oxidation of like 2 double bonds or
Okay.
2 cis bonds?
Sure.
The general picture of it was...
You don't like that picture, yeah. Nobody likes fatty acid oxidation of unsaturated ones. So the one you're talking about, I'm sorry, that's not that one. It's actually this one. Okay, this is the one you're talking about, right? Okay. This involves Cletus and Wallace. All right. There's nothing magical about having 2 double bonds. The reason we show you this with 2 double bonds is because the spacing gives us examples of both structures that are considered. Okay? So let's think about what happens during fatty acid oxidation of this compound. All right? Well, when I start with this is carbon number 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay? There's the carbon that has a cis bond. And we remember, of course, that for fatty acid oxidation, we have to have trans bond position 3, 4. So everything that's involved in both Cletus and Wallace here, is arranging so that we have a trans double bond at positions 2 and 3. That's all that's up with everything that you see on the screen. Okay? Positions, I'm sorry, I said 1, 2, yeah, 1, 2, 3. Okay. So we have in this case, we have a double bond that's in the cis configuration, and it's at positions 3 and 4. Okay? That's not going to work. All right? So, well, how do we get from here from this one down to this one? Well, we went through several rounds of beta oxidation, just clipping off 2 carbons at a time. There were no double bonds in the way, so the cell just clipped off 2 carbons, 2 carbons, 2 carbons. It got down to this structure, and now it's got a structure that it can't go any further unless it does something with. It's got a cis at positions 3 and 4. Cletus comes in, and Cletus does one thing, and it does one thing very well. It converts a cis at 3,4 to a trans 2,3. The trans 2,3 is exactly like the intermediate in beta oxidation. Remember, we first pull out those 2 hydrogens and electrons in that first step with the acyl-CoA dehydrogenase. It pulls them out between carbons 2 and 3. Makes a double bond, makes a trans double bond at position 2 and 3. Here's a trans double bond at position 2 and 3. Exactly what we have during regular beta oxidation. If I add water across this, as I do in regular beta oxidation, I put a hydroxyl right there. Right? Okay? So I get that hydroxyl. This figure is not showing you those steps. It's skipping those steps. I put a hydroxyl there. After I put the hydroxyl there, I oxidize that hydroxyl to a ketone. And then I do thiolytic cleavage, meaning I cut off those 2 carbons. Bang. And that's what I've done to get to here. Notice the blue and the blue. I've cut off these two carbons, just like I did in beta oxidation. Well, now I've got a structure that has a double bond way out here at 4 and 5. Okay? Way out here at 4 and 5. Beta oxidation starts. Okay? Beta oxidation starts because the acyl-CoA dehydrogenase can get in here and it can, in fact, remove those protons and electrons to make a 2,3 cis double bond, a 2,3 trans double bond, right? So we see that right here. However, the enzyme that would normally add water across this double bond can't do it. It doesn't like this structure because it doesn't like this bond right here. So that means that this structure has got to be reconciled so that the cell can, in fact, continue this oxidation process. Well, this is where Wallace comes in. Wallace is the enzyme that takes a trans 2,3 and a cis 4,5. It uses NADPH and makes a cis 3,4. This is the structure that was wrong in the book. Yes?
So do these double bonds have to be in this particular places that they are in order for this to work? Exactly.
They will be in that place.
Okay, but if in the beginning at lineoidal CoA,
Right.
if the cis's weren't together, if they were maybe a few carbons apart,
Okay.
it would be a different mechanism.
So her question is, well, what if the spacing on these cis's were different? Would this process work differently? And the answer is yes, but for the most part, they're not. When double bonds go into the polyunsaturates, they go 3 carbons apart. So in essence, the answer is no, they won't, although they could. Okay? So what I'm showing you here is what the cell is actually going to do. You're not going to see variation in this spacing. Okay? Okay. Well, again, Wallace takes 2 double bonds. It makes 1 double bond. That 1 double bond has a cis between 3 and 4. And not surprisingly, Cletus knows that very well. A cis between 3 and 4 going to a trans between 2 and 3 is what Cletus catalyzes, and Cletus takes care of that, and we're set. Yes?
So for acetyl-CoA carboxylase, I have it written down that it's inhibited by palmitoyl-CoA and palmitate. Is it both? Is it one or the other?
Yeah, I use the two interchangeably. So palmitoyl-CoA and palmitate, either answer is acceptable as far as I'm concerned. Okay? Did I answer the rest of your question okay? Okay. Any other questions on this? Because I know there's a lot of confusion for people. Cletus and Wallace. I can guarantee you, okay, there is a Cletus-Wallace question on the exam. And yes, Cletus and Wallace will be perfectly acceptable answer. Or if you want to give the full name, that's fine too. Yes, Jen.
How does the cobalt in the vitamin B12 cause free radical form when you're oxidizing others fatty acids?
Okay. I can't tell you how it does it. Okay. I can tell you that it does do it, and it's necessary for the movement of that methyl group, but I don't have an electronic mechanism for you to tell you for that. Yeah. This is the only place, I think I mentioned in class, it's the only place in biology that I'm aware of that we find cobalt doing anything. It's the only place it exists. I hear shuffling. Any other questions? Everybody decides they want out of here. Okay. So I will see you, if I don't see you at the review session on Thursday night, I will see you in here on Friday morning.